This can also be applied to antibody drug conjugates. Here I'm showing how we are implementing this technology to look at the conjugation of antibodies in the presence of all these chemicals that are used in the conjugation process. So there has to be, there does not have to be any type of cleanup. You can just simply do the analysis in the presence of all the other chemical or unconjugated species. So what's possible here is that we can definitely determine the titer of an ADC, which I'll show in a few minutes. But what we can potentially do in the future is to look at the conjugation efficiency by dialing in different types of reagents that are selectively binding to the drug itself, which is shown here. It's not currently available, and there's a thought that this could be used to look at the FRET from the first fluorescent tag to another reagent that binds to the payload. And this level of detection can give you specificity as well as look at the conjugation levels of this drug conjugate. So what I do want to share with you today is that we've also examined these antibody drug conjugates and measured their titer. So there are 3 different maps that I'm showing you here. Is that the first one is showing the conjugation of ADC, which had a DAR of 8. And this antibody sample was analyzed in the presence of all the other chemical conjugates that are present that are not bound to the antibody, and we see a single peak here. And in the bottom 2, we see the map 2 is bound to what is known as a radioconjugate, a DOTA molecule, which is used in nuclear medicine. And the third one is a very popular antibody drug conjugate called MMAF.